Tardigrades, small creatures famous for their toughness, have amazed scientists for years. These little beings can survive extreme conditions, but their secret to avoiding death has remained a mystery. Now, researchers are revealing the surprising reasons behind their incredible ability to live on. What new discoveries will we find, and how might they change what we know about life? Tardigrades, popularly known as water bears, are small micro-animals that measure less than 1 millimeter in length. Despite their size, they show remarkable flexibility, thriving in a variety of habitats ranging from the deep ocean to high mountain peaks and harsh environments such as Arctic tundras and hot springs. Johann August Ephraim Gutzi, a German biologist, described the unusual species of the Tardigrade phylum in 1773. One of the most notable features of tardigrades is their 8 legs, each having tiny claws that support them in navigating different surfaces. Their segmented bodies are coated in a protective cuticle that is shed on a regular basis to allow for development. This anatomical structure not only facilitates mobility, but also serves as a protective barrier against severe environmental conditions. Instead of swimming or wriggling, this little organism uses its short legs to move across microalgae. Tardigrada, which means slow walker, was later named by Italian scientists Lazzaro Spallanzani and Giovanni Battista in 1776. Interest in tardigrades grew, particularly in 1842, when a French zoologist, Louis P. de Règne, observed these microorganisms in their natural habitats, especially in moist algae and lichen. During an experiment, he mistakenly dried up their environment, causing them to move much more slowly. Later, he couldn't find them and assumed they were dead. When he added water to the sample, he was surprised to see the tardigrades return to life. This event caused him to reconsider his idea of life and death in creatures. De Règne's research led to the discovery that tardigrades live in a state of cryptobiosis, in which metabolic activity decreases to practically undetectable levels, allowing them to survive harsh circumstances such as dehydration, strong radiation, and freezing temperatures. Their metabolism can slow down to 0.01% of normal, allowing them to survive on a very small amount of food, much less than what a person consumes in a day. This unique ability to enter a state of suspended movement helps them protect themselves from extreme environmental changes, but it also raises concerns over the systems that allow such survival. Several studies have been carried out to investigate how tardigrades survive lethal amounts of radiation and cellular damage caused by the formation of ice crystals within the cells. So, how are tardigrade molecules revolutionizing drug stability in harsh environments? According to an innovative study from 2017, Thomas Boothby and his colleagues at the University of North Carolina attempted to solve the mystery of how tardigrades manage to survive under harsh conditions. These microscopic organisms can withstand severe colds as low as -270 degrees Celsius and intense radiation exposure. As most living organisms are composed of 60 to 80% water, scientists were interested in studying the adaptations that allow these species to survive in extremely cold conditions. Many species have adaptations to survive in harsh environments, but these adaptations often fail under extreme conditions. Tardigrades show remarkable resilience. They can tolerate mild dehydration, cold temperatures, and even survive in conditions where atomic movement was completely stopped. They gradually lose water content, which can reach up to 97%. The slow loss of water minimizes the formation of ice crystals within their cells. Tardigrades adapt by contracting their bodies to reduce surface area and exposure to harmful environmental factors. Researchers studied the molecular changes during this dehydrated state and revealed that tardigrades produce a novel class of proteins known as intrinsically disordered proteins when the tardigrade faces stressful conditions, protecting cellular structures like a shield. To verify the role of TDPs in survival, the research team inserted the genes responsible for these proteins into yeast and bacteria, which typically cannot survive in lost moisture. The modified organisms exhibited remarkable toughness to extreme drying, confirming that TDPs create a protective barrier similar to that of tardigrades. However, they do not prevent DNA damage from ionizing radiation, highlighting the challenges of survival in harsh environments. Most living things, including humans, are easily harmed by radiation, but tardigrades are different. They can survive extremely high levels of radiation that would be deadly to most other organisms. The median lethal dose, LD50 for humans, which is the amount of radiation that may kill 50% of people, varies between 2.5 and 4.5 gray. Tardigrades are resistant to radiation doses up to 4,000 gray. A team of researchers at the University of North Carolina at Chapel Hill, led by biologist Bob Gold, focused on these extraordinary features. The study focused on the tardigrade species Hypsebius exemplaris, which was chosen for its hardness and unusual biological features, making it an ideal model organism for research. The researchers exposed tardigrades to high doses of gamma radiation, far exceeding levels that would be lethal for humans. They used advanced genomic sequencing and proteomics to study the molecular responses of the tardigrades to this radiation damage. The results showed that instead of just destroying damaged cells, the tardigrades' exposure to radiation caused hundreds of genes to be activated, which make proteins for DNA repair and protection. This discovery highlighted tardigrades' capacity to adapt successfully to intense stress at a molecular level. Two key proteins, Dsup and TRD1, play important roles in protecting tardigrades from radiation damage. Dsup binds directly to DNA, forming a barrier that shields it from harm and reduces the occurrence of double-stranded breaks. TRD1 helps stabilize chromosomes under stress by acting like molecular glue, allowing normal repair processes. Together, these proteins highlight their specialized mechanisms for surviving radiation. Tardigrade biomolecules are being used as innovative products in space exploration and medicine. NASA's upcoming missions, including the Artemis program, face the challenge of cosmic radiation, which threatens astronauts' DNA. A more advanced approach involves engineering tardigrade molecules like TRD1 into human genes to boost DNA repair mechanisms. Recent discoveries of a tardigrade species that can emit light to survive UV radiation suggests the possibility of creating synthetic UV protection, which could further improve astronauts' safety in space. In the medical field, tardigrade biomolecules are being explored for their ability to stabilize life-saving drugs and protect transplanted organs. Researchers at the University of Wyoming are working on applying these molecules to enhance the transport and effectiveness of essential drugs in remote or harsh environments. If you like this video, don't forget to like and hit the subscribe button and get updated with more amazing content on our channel.